Hi there, this is the first in a series of videos I'm going to do on enzymes. If you are following along at home with your notes, this is on page 17 in the Biological Molecules booklet, so booklet 1 in the first year. Okay, so this is just really an introduction to enzymes, how they function, basically what they are and what they do. And then we will spend a bit more time on these two models of enzyme action. So two explanations of how enzymes operate. Okay, so just as an introduction, they are globular proteins. They have a three-dimensional shape, tend to be soluble. The importance of their tertiary structure, well, their tertiary structure is very important and it is specific. Okay, we'll come back to that shortly. They act as biological catalysts, so they are responsible for catalyzing or allowing reactions to happen inside cells and outside cells. And it is those reactions which will determine the structure and function of both cells and organisms, and the whole organism. Okay, they act as catalysts by lowering the activation energy. So we'll look at this concept of activation energy shortly. But in summary, activation energy is the energy required for a reaction to take place. So energy required for a reaction to take place. Now, in biology, often reactions are taking place at quite low temperatures. And at those low temperatures, those reactions would not proceed quickly or at all. So it's necessary to have these biological catalysts, the enzymes, to allow those reactions to occur. Inside cells, inside organisms, there are hundreds of thousands, tens of thousands of reactions that are taking place. Almost every one of them will be catalyzed by an enzyme of some description, and maybe some biological pathways, metabolic pathways, series of reactions may involve a series of different enzymes. They are soluble in water, as I said earlier on, due to their hydrophilic side groups. So side groups, those R groups on the polypeptide chains that face outwards. So the amino acids have those R groups which are polar. Those polar R groups interact with water. We refer to them as being hydrophilic, and they face outwards. Any hydrophobic R groups tend to face inwards, fold inwards on the protein, so they would form the internal parts of the protein. Now, these two words, anabolic and catabolic, you don't need to know them, they're not on the specification, but you may have heard of them. And they summarize really the types of reactions. So that some enzymes catalyze anabolic reactions, i.e. taking smaller molecules and making them into larger ones, whereas other enzymes do the opposite. So they take larger molecules and make them smaller. Like I say, you don't need to know those, but you may be familiar, or you may have read those terms round and about. Okay, so the important feature of an enzyme and its tertiary structure is that tertiary structure produces an active site. So you may be familiar with this from when you did your GCSEs. The active site is a small hollow depression made up of just a small number of amino acids, just a few amino acids, in the overall three-dimensional structure of the protein, of the enzyme. And it is that active site which is complementary to a substrate. And when I say substrate there, or complementary to a substrate, we mean that they are specific. So, enzymes may be specific to a single substrate. For example, catalase is complementary to the shape of hydrogen peroxide. Only one substrate is able to fit in that particular active site. So they are complementary, and I cannot emphasize how important this word is. Other enzymes may be specific to a particular bond. So lipases are specific to the ester bonds that form between glycerol and fatty acids. Okay, that specificity is produced by the tertiary structure of the protein. So obviously all enzymes are three-dimensional proteins. They have a tertiary structure determined by the position of hydrogen, ionic, sometimes disulfide bonds in that tertiary structure. That gives a specific shape and electrostatic charge to the active site, that hollow depression that is complementary to the substrate. Okay, an important thing to note here is that the active site and substrate are complementary but not the same shape. Okay? Regardless of how many times your biology teachers will tell you this, there'll always be somebody who says they are the same shape. It comes up in the exam series every year. They are complementary but not the same. Like a hand fitting into a glove. Okay, so here is a diagram of an active site. The active site shown by the this polypeptide chain with these R groups pointing out of the polypeptide chain. So those R groups and, in fact, the amino group from the amino acid itself allows for the formation of, well, hydrogen bonds here. There's an ionic bond there. So those interactions between the substrate, so the substrate here is this molecule, and it interacts in a very specific way with the shape of the active site. Okay, and as it does that, those temporary hydrogen ionic bonds form, then the reaction takes place, and bonds will either be formed between two substrates or broken within the substrate. Now, there are two methods, or two models, which explain that we'll come back to shortly. But before we do that, I mentioned earlier on this idea of activation energy. So if you read in your notes, the activation energy is the energy required for chemical reactions to take place. Enzymes are able to lower the activation energy, meaning less energy is required for those chemical reactions to take place. And the importance of that is that biological systems tend to work at low temperatures. So there isn't a lot of energy available in the environment where these reactions are taking place, so instead they require an enzyme to help with that catalysis, to help with the reaction. The enzymes lower the activation energy through the formation of enzyme substrate complexes. This is an enzyme substrate complex, and enzyme substrate complex, along with complementary, along with the types of bonds like ionic, hydrogen, disulfide bonds. These are terms that are really vital when answering questions on in A-level biology. If your response to a question doesn't on enzymes doesn't include a term enzyme substrate complex, you need to go back through and put it in there. Make sure it's in the right context, but it should be there. Okay, so by lowering the activation energy, it is possible for those enzyme substrate complexes to form more frequently. So there's an increase in the rate of the formation of these enzyme substrate complexes, and therefore the reactions will take place more rapidly. So, two models for explaining enzymes. The first is one that you've probably seen before at GCSE. This is called the lock and key model, lock and key principle, where a substrate with a particular shape is complementary to the active site of the enzyme. As that substrate enters the active site, we get the formation of that enzyme substrate complex. At this stage is where the catalysis takes place. So that substrate becomes a product. The products then leave the active site, and the enzyme can be reused and can accept a new substrate. Okay, so during this period of the enzyme substrate complex, those temporary bonds form between the amino acids and the substrate molecule, just as we can see here. Okay, but this lock and key principle, lock and key model, is limited. Okay, it doesn't really explain how the activation energy is lowered. And at A level, it's necessary to go beyond this lock and key model to use an improved model, which provides us with some a better understanding of how the activation energy is lowered. And this improved model is referred to as the induced fit model. In the induced fit model, the active site of the enzyme is not complementary to the substrate, or initially, before the reaction, is not complementary to the substrate. As the substrate interacts with the active site becomes more complementary. The active site changes shape, becoming more complementary as the enzyme substrate complex is formed. Remember, during that period when the enzyme substrate complex is formed, that's when those temporary hydrogen ionic, perhaps the hydrogen and ionic bonds form between the substrate and the active site. Okay, that changing of shape of the active site stresses bonds in the substrate molecule. So it applies stress to those bonds, meaning that the energy required to break those bonds is reduced. This leads back to the activation energy. So we can see here the activation energy with an enzyme is lower than the activation energy without the enzyme. The reason for that, the substrate enters the active site, which changes shape. As it changes shape, that puts strain or stress or distorts the bonds inside the substrate. That makes it easier for those bonds inside the substrate to be broken, meaning that the reactions are able to take place at a lower temperature. Then the products are formed. As the products leave the active site returns to that initial shape that is not complementary to the substrate. This enzyme, with its non-complementary active site, can then bind to another substrate, and the process is repeated. This is shown again in this diagram. So we can see here the active site is not complementary, but as the substrates enter, the active site becomes more complementary. And in this case, the active site becoming more complementary brings these two substrates together, making it easier for bonds to form. So we've got two concepts here. Either there is stress on the bonds in the substrate molecule, making them easier to break, and perhaps bringing two groups together closer in proximity, closer in space, that might make it easier for a bond to form. Both of those concepts provides us with an explanation as to how this activation energy is lowered. Okay, so the enzyme substrate complex is formed. The active site becomes complementary as that enzyme substrate complex is formed. Then the products are produced and released, and the active site returns to the non-complementary shape. Here's a question taken from an exam. 2 marks, describe the induced fit model of enzyme action. So we've got the mark scheme down here. So before the reaction, we know that the active site is not complementary. Okay, and we can see that in this induced fit model diagram and in this one. Okay, so before the reaction, not complementary. As the substrate binds, the active site becomes complementary. And then the enzyme substrate complex is formed. I'm using an abbreviation here, ES complex. Now on some mark schemes that may be sufficient. My advice would be to play it safe. Don't be lazy like I'm being now, and actually write enzyme substrate complex. Okay, so the enzyme substrate complex is formed. Now that would be enough for the 2 marks, but there was a 3rd mark available in this question, and that 3rd mark was that the formation of the enzyme substrate complex stresses the bonds within the substrate. Okay. Which leads to a reaction. Okay, so actually those first two marks are more of a description. If it were an explain question, perhaps explaining how the induced fit model lowers the activation energy, you would need to include this third point here, that the as the substrate enters the active site becomes complementary, enzyme substrate complex is formed, which puts stress or strain or distortion on the bonds within the substrate, leading to the reaction. In fact, I think for that third mark, if you'd said that it brings the groups within the substrates closer together, leading to the reaction, that would also be acceptable there. Okie doke. I'm going to leave it there for the first part of our introduction to enzymes. So the key points here are the induced fit model as an improved explanation for how enzymes lower the activation energy. Okay, thanks a lot.